Science Quest, Episode 1 Unlocking the Universe, CRISPR, Gene Editing, and the Future of Medicine. Did you know that scientists can now edit our genetic code with the precision of a word processor? Imagine a world where genetic diseases are eradicated, where we can enhance our own capabilities, where we rewrite the very blueprint of life. Welcome to Science Quest, where we explore the cutting edge of scientific discovery. Today, we're diving deep into the revolutionary world of CRISPR-Cas9 gene editing. Our DNA, the instruction manual for life, is made up of billions of base pairs. Think of them as letters in a code. Genes are specific sections of this code that determine our traits, from eye color to susceptibility to disease. For decades, gene editing was a dream, a futuristic fantasy. But then came CRISPR. CRISPR-Cas9 is like a pair of molecular scissors, incredibly precise and programmable. It's a naturally occurring system found in bacteria, adapted by scientists to target and cut specific DNA sequences. Imagine you have a typo in your instruction manual. CRISPR can find that typo, cut it out, and replace it with the correct sequence. The magic lies in the guide RNA, a short strand of RNA that acts as a homing device, guiding the Cas9 enzyme to the precise location on the DNA strand where the edit needs to be made. This allows us to correct genetic errors, insert new genes, or even switch genes on or off. The potential applications of CRISPR are mind-blowing. We're already seeing progress in treating inherited diseases like sickle cell anemia and cystic fibrosis. Imagine a future where these debilitating conditions are a thing of the past. CRISPR isn't just about human health, it's revolutionizing agriculture by creating disease-resistant crops, boosting yields, and reducing the need for pesticides. It's even showing promise in fighting infectious diseases like HIV and malaria. But with great power comes great responsibility. Ethical concerns surrounding CRISPR are crucial. The potential for unintended consequences, off-target edits, and germline editing changes that are passed down to future generations raises serious questions about safety and societal impact. Scientists are working tirelessly to address these concerns, developing more precise and safer CRISPR systems. We're at a critical juncture. The power to edit the human genome is in our hands. It's a powerful tool that needs careful consideration and responsible stewardship. We must engage in open dialogue, establishing ethical guidelines and regulations to ensure that this incredible technology is used for the betterment of humanity. The future of CRISPR is bright. As scientists refine the technology, we can expect even more groundbreaking applications. Imagine personalized medicine tailored to your individual genetic code, or the eradication of diseases that have plagued humanity for centuries. The journey has just begun, and the possibilities are limitless. We've explored the revolutionary science of CRISPR-Cas9 gene editing, its incredible potential, and the vital ethical considerations that must guide its development. But next time on Science Quest, we'll be delving into the exciting world of fusion energy, the power source of stars, now within our reach. Join us as we explore how this groundbreaking technology could revolutionize energy production and combat climate change. Science Quest, Unlocking the Universe, CRISPR Gene Editing. Did you know that we're on the verge of writing the very code of life? Forget sci-fi fantasies. We're talking about CRISPR, a gene editing technology so precise, so powerful, it's revolutionizing medicine, agriculture, and even our understanding of evolution itself. Welcome to Science Quest, where we explore the cutting edge of scientific discovery. Today, we're diving deep into the world of CRISPR and uncovering its incredible potential, as well as the ethical dilemmas it presents. CRISPR stands for clustered regularly into spaced short palindromic repeats. Sounds complicated, right? But the basic concept is surprisingly simple. Imagine DNA as a vast, intricate instruction manual for life. CRISPR is like a pair of incredibly precise molecular scissors guided by a GPS system. This GPS is a short RNA molecule that targets a specific sequence on the DNA. The scissors, the Cas9 enzyme, then cuts the DNA at that precise location. This cut allows scientists to either remove a faulty gene, insert a new one, or even modify an existing gene. Think of it as editing a word processing document, but at the level of our genes. The potential applications of CRISPR are staggering. In medicine, we're talking about curing genetic diseases like cystic fibrosis, sickle cell anemia, and even certain types of cancer. Imagine a future where inherited diseases are a thing of the past. Beyond human health, CRISPR offers enormous potential in agriculture. We can engineer crops to be more resistant to pests, diseases, and even climate change, leading to increased food production and reduced reliance on pesticides. We could even enhance nutritional value, making food healthier and more sustainable. But with such incredible power comes great responsibility. CRISPR raises profound ethical questions. Should we be editing the human germline, making changes that are heritable and passed down through generations? What are the potential unintended consequences? Who decides which genes are desirable and which are not? These are complex issues with no easy answers. It's crucial to have open and transparent discussions involving scientists, ethicists, policymakers, and the public to navigate these challenges responsibly. The research on CRISPR is constantly evolving. Scientists are exploring more sophisticated applications, including targeted drug delivery, personalized medicine tailored to an individual's genetic makeup, and even regenerative medicine, where damaged tissues and organs could be repaired using CRISPR-edited cells. CRISPR gene editing represents a paradigm shift in our ability to manipulate life itself. It offers incredible potential to address some of humanity's most pressing challenges, from disease to food security. However, it also forces us to confront complex ethical dilemmas that require careful consideration and responsible action. The future of CRISPR depends on our ability to harness its power wisely and ethically. Next time on Science Quest, we'll explore the mind-bending world of quantum computing, the technology that could revolutionize everything from medicine to artificial intelligence. Prepare to have your perceptions of reality challenged. Science Quest, Unlocking the Secrets of the Universe, Episode 1, CRISPR Gene Editing Revolution. Welcome back to Science Quest, where we explore the mind-blowing advancements shaping our future. Did you know that scientists can now edit our genes with the precision of a word processor? This isn't science fiction, it's the reality of CRISPR, a gene editing technology poised to revolutionize medicine and beyond. CRISPR-Cas9 is like a highly advanced molecular scissors and paste tool. Imagine DNA as a long instruction manual for building and running a living organism. Sometimes there are typos, genetic mutations that cause diseases. CRISPR lets us go in, find those typos, and correct them or even rewrite sections of the manual. It's based on a natural defense mechanism found in bacteria, which use it to fight off viruses. Scientists have harnessed this powerful system to precisely target and modify specific genes in virtually any organism. CRISPR stands for Clustered Regularly Interspaced Short Palindromic Repeats, quite a mouthful. But the key player is the Cas9 enzyme, which acts like the molecular scissor. Scientists design a short RNA sequence, a guide RNA that matches the specific DNA sequence they want to target. This guide RNA acts like a GPS, leading the Cas9 enzyme to the precise location on the DNA strand. Once there, Cas9 cuts the DNA, allowing scientists to either disable the gene or insert a new gene sequence. The implications of CRISPR are staggering. Imagine a future where we can sickle cell anemia, cystic fibrosis, Huntington's disease. These devastating conditions could potentially be eradicated by correcting the faulty genes responsible. CRISPR could be used to engineer immune cells to more effectively target and destroy cancer cells. By modifying genes, we can create crops that are more resistant to pests, diseases, and harsh environmental conditions. CRISPR can help modify bacteria to make them more susceptible to antibiotics, combating the growing threat of antibiotic-resistant infections. Despite its immense potential, CRISPR raises significant ethical concerns. The power to edit the human germline, the genes passed down to future generations, raises questions about unintended consequences and the potential for designer babies. There are also ongoing debates about accessibility, affordability, and the potential misuse of this technology. Rigorous safety testing and careful ethical guidelines are crucial to ensure the responsible development and application of CRISPR. CRISPR technology is rapidly evolving. Scientists are constantly improving its precision and efficiency, exploring new applications, and developing safer and more accessible methods. The future holds immense promise for CRISPR, offering the potential to transform healthcare, agriculture, and even our understanding of life itself. CRISPR-Cas9 represents a paradigm shift in biotechnology, giving us unprecedented power to manipulate the building blocks of life. While ethical considerations are paramount, the potential benefits of this revolutionary technology are too significant to ignore. In our next episode of Science Quest, we'll dive into the exciting world of quantum computing and get ready to explore the possibilities of a computational revolution. Science Quest, Unlocking the Universe Episode 1, CRISPR, the Gene Editing Revolution. Ever wish you could edit your genetic code like a word processor? Sounds like science fiction, right? Well, today, we're diving headfirst into the incredible world of CRISPR-Cas9, a revolutionary gene editing technology that's rewriting the rules of biology and sparking a global conversation about the future of medicine, agriculture, and even humanity itself. Forget clunky, imprecise gene editing tools of the past. CRISPR is like having a pair of incredibly precise molecular scissors guided by a GPS system. This system, naturally occurring in bacteria, uses a guide RNA molecule to target a specific DNA sequence. Think of the guide RNA as a search engine, locating the exact spot in your DNA that needs editing. Once found, the Cas9 enzyme, our molecular scissor, cuts the DNA at that precise location. This allows scientists to insert, delete, or replace genes with remarkable accuracy. This seemingly simple process holds immense potential. Imagine a world where genetic diseases like cystic fibrosis, sickle cell anemia, and Huntington's disease are things of the past. CRISPR is already showing promise in clinical trials, offering the possibility of curing these debilitating conditions. But CRISPR isn't just about human health. Its applications extend far beyond the clinic. In agriculture, CRISPR is being used to develop crops that are more resistant to pests, diseases, and harsh environmental conditions. This could revolutionize food production, ensuring food security for a growing global population. Imagine drought-resistant wheat, pest-resistant corn, and nutrient-rich rice, all thanks to CRISPR. Now, let's talk about the elephant in the room, the ethical considerations. The power to edit the human genome is a profound responsibility. We need to carefully consider the potential implications of altering the human germline, the genes that are passed down from generation to generation. What are the potential unforeseen consequences? Should we be editing human embryos? These are complex questions with no easy answers, and they require open and honest public discourse. One concern is the potential for off-target effects, where CRISPR accidentally edits the wrong part of the genome. Scientists are working hard to minimize these risks, improving the accuracy and specificity of the CRISPR system. Another concern is the accessibility of this technology. Will it only be available to the wealthy, exacerbating existing health inequalities? Ensuring equitable access is crucial. Despite these ethical challenges, the potential benefits of CRISPR are undeniable. It represents a paradigm shift in our ability to understand and manipulate life itself. We are standing at the cusp of a new era in biology and medicine, an era shaped by our ability to edit the very blueprint of life. But the journey doesn't stop here. Scientists are continually refining CRISPR, exploring new applications, and addressing the ethical challenges. Next week on Science Quest, we'll delve into the exciting world of fusion energy, a potential solution to our energy crisis. Will we soon be harnessing the power of the sun on Earth? Join us to find out. Science Quest, Unlocking the Universe Episode 1, The CRISPR Revolution and Gene Editing. Imagine a world where genetic diseases like cystic fibrosis, Huntington's disease, and even certain types of cancer are a thing of the past. Sounds like science fiction? Think again. Today on Science Quest, we delve into the groundbreaking world of CRISPR-Cas9, the gene editing technology revolutionizing medicine and beyond. CRISPR, short for clustered regularly interspaced short palindromic repeats, isn't some alien technology, it's actually a naturally occurring defense mechanism found in bacteria. These bacteria use CRISPR to fight off viral infections by remembering the virus's DNA and cutting it up when it attacks again. Scientists cleverly harness this natural process, creating a precise gene editing tool. Think of it like molecular scissors, allowing us to snip out faulty genes and replace them with healthy ones. The Cas9 part is an enzyme that acts like the blade of these molecular scissors, guided by a specific RNA sequence that targets the DNA we want to edit. The potential applications of CRISPR are vast and breathtaking. Imagine a future where we can eradicate inherited diseases before a child is even born. This is no longer science fiction. Clinical trials are already underway for a range of conditions, including sickle cell anemia and beta thalassemia, both debilitating blood disorders. CRISPR is also showing promise in the fight against cancer, allowing scientists to engineer immune cells to more effectively target and destroy cancer cells. Beyond human health, CRISPR has applications in agriculture, creating disease-resistant crops and improving crop yields to address global food security challenges. While the potential benefits are enormous, CRISPR also raises significant ethical concerns. The ability to alter the human germ and the genes passed down from generation to generation opens a Pandora's box of possibilities, some potentially problematic. What are the long-term consequences of altering our genetic makeup? Who decides which genes should be edited? These are critical questions that require careful consideration and robust ethical frameworks. The scientific community is actively engaged in discussions about responsible gene editing guidelines and regulations to ensure this powerful technology is used ethically and responsibly. The field of CRISPR gene editing is rapidly evolving. Scientists are continually refining the technology to improve its accuracy and efficiency. Base editing, for instance, allows for more precise changes to individual DNA bases without causing a double-stranded break in the DNA, minimizing the risk of unintended consequences. Furthermore, researchers are exploring the use of CRISPR for gene therapy outside of the laboratory setting, developing methods for delivering CRISPR components directly to target tissues in the body. Let's test your knowledge. What are some potential benefits and risks of using CRISPR gene editing technology? Think about this for a moment, and then share your thoughts on our Science Quest social media platforms using CRISPR Quest. CRISPR-Cas9 represents a monumental leap forward in our ability to understand and manipulate life itself. While ethical considerations remain paramount, the potential to alleviate human suffering and advance our understanding of biology is undeniable. The next revolution in genetic engineering may well be just around the corner.